Morning, everybody. We're so pleased to have you with us for the symposium. So I'll just briefly explain how things work for those who weren't here today. We have 15-minute time blocks, so that means 5 minutes of presentation, about 5 minutes of question and answer, and then some transition time between presentations. So we are on Zoom so that family and friends can see the presentations, and we're recording also so that this can go in the portfolio for our scholars. So the room does pick up various noises, so we ask you to try to keep as quiet as possible, but otherwise to fully engage and think of great questions. And with that, I'll introduce our first presentation. I'm pleased to introduce Jane Arterberry, who's worked with Dr. Ken Stedman and his lab. And over to you to get started. Thank you.
Thank you. So I'm Jane. In the summer, I worked in the Extreme Virus Lab on a project focusing on localizing the structural proteins of SSV1 via site-directed mutagenesis. So an overview of what we study: SSV1, Saccharomyces spindle-shaped virus 1, is an archaeal extremophilic virus. It's found in high temperature, low pH environments like volcanic hot springs. And it is comprised of a number of structural proteins that contribute to its unique lemon shape. VP3 is thought to support the spindle. VP4 is thought to comprise the tail. And C166 is thought to comprise this intermediary region. And notably, all of these structural proteins lack cysteine residues, and their locations have yet to be confirmed. So this leads into what my specific project is looking at, which is creating cysteine mutations in the open reading frames of these proteins, electroporating them, culturing these mutant viruses, virions, concentrating the virions, and then visualizing them under TEM with this unique particle called nanogold. Nanogold binds specifically to cysteine residues, and since their structural proteins naturally lack it, it works perfectly to actually show us on the virions themselves where these proteins are found. So progress this summer has been slow but hopeful. That's science. Some ways we confirm successful mutagenesis and infection include halo assays and PCR. So here I have a halo assay showing the successful infection of our host with a VP3 mutant. As you can see, it's very faint on the screen, but we've got a clearing here compared to the 1% SDS control. So that's showing that we have a VP3 mutant we can look at, and a PCR of the C166 mutants. I have 2 that are thought to be mutants that lit up for the supernatant compared to the wild type and the wild type plasmid DNA. Now, with these mutants, we need to work on concentrating them and then finally visualizing them under the TEM, like this image here of wild type virions we imaged with vanadium negative stain, which is the stain that we will use alongside the nanogold. It provides a really dark background and makes the virions, you can see the little lemon shapes, stick out really well and appear a lot brighter, which will act as a really great contrast because nanogold appears as these tiny black dots, which I will show later. So some takeaways from this project thus far: getting mutant DNA into the cells and into the genomes is very, very challenging. It's been a bottleneck for me, but you just got to keep repeating it until you can get it in. Another thing we've learned is that the negative stain is compatible with SSV1 imaging. We learned that using our wild type, so we know we can move forward using that when we're actually ready to image the mutants. And another lesson we've learned is that AlphaFold is really great in helping us select mutagenesis sites because it can give us these predicted structures, as you can see here. And I used AlphaFold to be able to select some residues that are like equal distance away and hopefully showing out on the surface. That way we can actually see them and they're not accidentally internal. So some future directions for this project are pretty clear. We are definitely gonna look at visualizing the mutant virions under TEM, and we are focusing on producing and concentrating more of these mutant virions. And what we're shooting for is something similar to right here. These black dots are the nanogold. This is a phage. This is pulled from a completely different paper, but they're using it for the same purpose. This is under TEM. These are cysteine residues they're binding to. With that, I would like to thank my lab mates. I would like to thank everybody who's working on SSV1s. And I'd like to support or thank those supporting this research, like the NIH and Bill Nixito. So especially my two mentors.
Thanks.
Any questions? Yes.
You mentioned that the structural proteins didn't have cysteine residues, I think it was. Why is that important?
Because the nanogold only binds to cysteine residues. So we can use that as like elimination to make it so it only specifically binds to that. Yes.
So is there something about the lemon shape that helps that virus survive in the extreme environment?
You know, I'm actually not sure. I know that that is something that is a question, I suppose, and we know which proteins are contributing to it. That's a better question, unfortunately, for like my mentor. But it's unique, that's for sure. Yes.
I loved how you shared some of the lessons about doing the work, you know, how it's hard, you gotta be persistent, and you're learning about the process as you go. Are those things that occurred to you, or did the mentor point those out, or how, I mean, it's great that you shared with us.
It's definitely something that I've learned, and I've also learned from my mentors. I know that last week, you know, when everybody was scrambling to figure out like what they were gonna put in, I asked Jono, who's one of my mentors, like, does the disappointment of science ever go away? Like when it's wrong? And he said, like, no. But it's like part of it. I was like, okay, well.
So that's what I loved. You brought the transparency to the
Thank you.
presentation.
Yeah.
So.
I had a question that was along those lines, but it also sounds like you learned a lot of skills. So this is not my discipline. So there's a lot of words and things that came up here for me that I have questions about. For example, AlphaFold. Can you tell us a little bit about that?
Yeah.
Software and how you got to use it and learn about it?
Yeah, I think it's a relatively new software and it uses AI, which I don't understand very well, but I guess it takes the sequence as well as some of the other characteristics when you like download the genome and it shows us this prediction. It takes a long time, like multiple hours to run just to get one of these. And it's fairly limited. I know on Benchling, it's still in the like beta stage. But this was the first, at least for C166, this was the first time we could actually see what it might look like. And it is a prediction, but
That's cutting edge. It's cool.
yeah, it's exciting.
So I have a obnoxious question, is it EI? Kind of following up on the first question. Do you have any speculation why there are no cysteines in the normal sequence?
Yeah, I know we did talk about this and like the genome, you know, SSV1 is thought to be kind of like a marriage of two different organisms that is still not quite understood, but like the entirety of like the structural portion of SSV1 lacks cysteines, but like the other parts, it's completely normal and has cysteine, so. It's thought to be significant in that way, like it merged, this lacked it, this has it. But. Yeah, you could have said it better, but
No, great.
Yeah.
yeah, so. Thank you.
Thank you.
Yeah.
So, let's get started with our next presentation. Again, glad to have you all here. So, next we get to hear from Kylee Brevick, who is also a Build Exito Cohort 7 scholar, working with Dr. Ann Thompson. And I don't even attempt to talk about the titles because I can't pronounce it. Here we go.
Okay, thanks for the introduction. So yeah, I'll be telling you about my research on how I discovered picophytoplankton in both the Columbia and Willamette rivers. Okay, so I'll start by briefly going over the biogeochemistry of the system that I'm interested in. Well, I wanted to use this figure to emphasize the interconnectedness of phytoplankton. I'll focus your attention to the top portion of the image that shows how important these photosynthetic microorganisms are in fixing inorganic carbon and producing oxygen using sunlight, and also serving as a food source for higher trophic organisms. And so this makes phytoplankton key in determining energy, carbon, and other nutrient flows in aquatic ecosystems. More specifically, I study the tiniest of these photosynthetic cells, referred to as picophytoplankton, and majority of which are picocyanobacteria, shown in these images here. And so these cells are defined as being less than 2 microns in diameter and come in a variety of beautiful colors, shown in these concentrated cultures. And these colors are composed of the different pigments that these cells use to harvest sunlight and thus perform photosynthesis. And so these colors can even fluoresce using special instruments shown here. And so that's how we choose to characterize these cells based on the different types of sunlight that they're able to harvest. So with this project, I aim to answer the following. First, how many different groups or populations of picophytoplankton are present in both the Columbia and Willamette rivers? And then how do these population abundances change over space as well as time? And from there, my goal is to piece together the ecological relationship to understand how these cells and the environment shape one another. So to answer these questions, I sampled surface water at both the Columbia and Willamette, shown here on the map. I chose these sample sites based on there being nearby USGS stations that I can get high hydrological data from to use later in my analysis. And then I analyzed these surface water samples using a flow cytometer. This instrument's perfect for analyzing picophytoplankton because the hydrodynamic focusing of the water sample allows for the analysis of an individual cell at a time. Analysis involves a laser exciting each cell, and then the relative size gets detected, as well as pigment fluorescence like chlorophyll. And so in this way, I'm able to quantify and characterize all of the cells present in each sample. And so the data from the flow cytometer looks something like this. Here I show you 2 plots representing the same sample that I took from the Columbia this July. And so we are plotting the cells based on different parameters. So the cells are the dots on the plot, represented or color coded by the populations, the different populations that I found shown here. And so we start analysis by looking at chlorophyll fluorescence, because this is the pigment required for photosynthesis. And then from there, we analyze these cells based on an accessory pigment called phycoerythrin. And so in this way, you can see how the populations are different from one another based on the pigments. For example, C1, the light purple colored population, has a distinct chlorophyll ratio here that it shares with the light green population. However, these two are different in that if we look at phycoerythrin, C1 has little to no phycoerythrin. just an example there, and then in this way I can quantify all of the populations present in each sample, which I show you here, using a heat map of the log scale of cells per milliliter over my sampling time frame from last fall to this July. And the warm colors represent a high abundance with the cool colors a low abundance. And so some key takeaways from this data. First, each of these 7 different populations were present in both the Columbia and Willamette rivers over the sampling time frame, which with each of the populations having its own unique pattern. And then despite them being unique, there are some similarities between the two. For example, C1 and C2, the top 2 rows of each panel, both increase as we reach July. And then the Columbia River has a higher concentration of cells overall throughout the year. And so with that, I can conclude that I discovered 7 distinct populations of picophytoplankton that were dynamic over space as well as time. All of this alluding to that these cells fill spectral niches in which they're able to coexist by taking advantage of different wavelengths of sunlight. My next steps include to track the change in size of these cells, to perform statistical analysis on the USGS data, and then to eventually genetically identify these cells. With that, I will thank all of the funding resources as well as my amazing mentors and colleagues that helped me with this project. And any questions?
Yes, I was curious about the flow cytometry data when you're identifying these different populations. You have the colors, you have the beautiful sort of separation here of all these things. So how do you actually do that identification? Are these based on what's already known, or because I see sort of just some purple dots in the green dots and that kind of thing? And so how is that? How are you doing that?
Yeah, so these first we look at chlorophyll, like I said, so we start there, and then from there so I was able to first gate this group, and then from there differentiated between these two based on this relative level of pigment fluorescence.
Okay, so you gate on that one group there.
Yep.
And then go to the other channel and look in there, and then you can gate between those two, and then go back and label them later.
Yep.
So they're not coming out as different colors, you just see the dots, of course, in the flow cytometry.
Yeah.
It's a nice way of representing it.
It's all using a software called FlowJump.
From Ashland, Oregon.
Yes.
How does your work compare to other work that's been done on these cells in these in the Willamette and Columbia?
So it was expected that these cells are present in the river because we know they're in the ocean and in lakes. However, this is the first time anyone's ever distinguished between these populations in these rivers. So it's really exciting first study to do this. So I'm excited about that. Thank you. Yes.
So you chose these sites because they're co-located with USGS gauge information. What type of information are you interested in comparing your results to?
Yeah, definitely temperature. So I can get a sense of seasonal changes, and temperature just plays a big role in general. And then other data that I have available, turbidity, which plays a role with how much sunlight the cells can get, nitrate levels, a key nutrient, and then discharge up the river just to understand the velocity. I think that's about it. Yeah.
So for example, it looks like C1, 2, and 3 are more prevalent in the summer.
Yes.
So what might be going on there?
I think so. Here we can look at some of the data that I have so far. So I didn't go over this just because I still need to do some statistical analysis, but yeah, the turbidity drops off. That could be one thing. Also, the temperature increases a lot as we hit summer, so that's expected to increase productivity. Yeah, just some ideas. Yeah, Marcel.
Where exactly are these picophytoplankton located in the river? Are they closer to the shore? Are they further out? Or are they all out?
All throughout. So I've been sampling off a dock, so I tend to sample so it's less obstructed from anything, so open flow, but the river's flowing, so they're throughout. Mostly, I was talking with Dr. Pan recently, and mostly they're gonna be found on the surface layer just because of the turbidity of rivers. Doesn't really allow for much to grow too deep. So sampling the surface water is pretty representative.
So
Yeah.
for less technical people, is it technical as a takeaway, like this is a healthy level of life forms that you'd expect there? I mean, what does it tell us overall about how the rivers are doing?
Can't quite extrapolate that information yet. I'm setting the ground basis of what's even present to begin with. And so from here, once I do some statistical analysis on the data, I'll be able to present more significance as far as that relationship, but it's not good or bad. Let's just stick with that, yeah.
So it's not concerning.
Yeah, not concerning. These guys are not the cyano producing ones that we're used to hearing about. So not known to be toxic.
So kind of a follow-up question
Yeah.
to exactly that, which is, does any of your data overlap with these, toxic cyanobacterial blooms or not?
I'm curious, not sure. I'd love to look into that.
I was trying to remember because I think it was, this summer. that we
Yeah,
had
I going to
those, but-
sample, there's signs saying. be
Yeah,
careful.
and your data's through July, but I couldn't remember when they were, and that was July of this year, your data?
Yeah, and I have some samples from August that I need to process, and that's when the bloom really hit in the
Wow.
Willamette, so that'll be interesting for sure, yeah, totally.
Are you
Yes.
expecting all these organisms to be prokaryotes?
Not quite, good question. Let's see, like, for example, looking at this, all of the ones with CP are more than surely prokaryotes, specifically Synechococcus, but this group. Actually, it might be the red. There's one population here that we're pretty sure is pico-eukaryotes, just to be clear. So that's why I said pico-phytoplankton, just to state general, majority of which are, cyanobacteria. What else?
Thank you so
Thank
much.
you.
She's also preventing.
Okay, so go on to the next presentation. I think the word is getting out that we're having great presentations in this room. So we're excited to introduce Michelle Hesek, another Build Exito scholar who's been working with Dr. Alyssa Starry. And again, what's great about this program is it's such a diverse range of research topics. So interesting presentation. Thank you.
Thank you for the introduction. Good morning, everyone. My name is Michelle Hesek. I'm an environmental science major who is working for the Home Ecology Research Lab with Dr. Alyssa Starry. Today, I'll be talking to you about my presentation on how plant growth impacts weed coverage in experimental eco-roofs. Eco-roofs reduce stormwater runoff from impervious surfaces and the combined sewer system. Portland is known to have a combined sewer system that includes domestic as well as industrial sewage, as well as rainfall runoff. During times of excess flow, this runoff is discharged into natural water bodies such as the Willamette River. One way that we reduce stormwater runoff is by promoting green infrastructure, including eco-roofs. And the main component that absorbs water from the stormwater runoff is the plants. And a study even found that the plant biomass possibly impacts their retention capacity. And so the main question that we want to answer with this research is, how does plant growth impact stormwater runoff on eco-roof trays? In addition, we would like to study how the plant growth could impact the composition of the weed species within the trays. Maintenance is very important on eco-roofs, and a lot of people consider the weed species to be a nuisance. However, there have been some studies that found that they can actually provide a nutritional benefit to the substrate and the plants themselves. The main significance of this research is to, one, maximize the benefits for eco-roof owners, two, relieve some stress from the overwhelmed stormwater systems, and also third, be the first to provide long-term seasonal leaf area index data. Leaf area index is just the ratio of the plant canopy to the rest of the ecosystem, or in this case, the trays. It is also used to characterize the plant growth over time. Here is the structure design of our eco roof trays. They are located on the Walk of the Heroines. They are 16 feet squared in area. And we are using two of the most commonly planted eco roof plants, so buffalo grass and white stone crop. We have 7 structures in total. 3 are planted with the buffalo grass, 3 with the white stone crop, and 1 is used as our control with only substrate. Here is a side profile of the structure design. Every eco roof is equipped with a waterproofing membrane, which is just a thick layer of plastic that prevents any natural elements and also root penetration from getting to the roof surface, or in this case, the trays. There's also a filter fabric, which allows any excess water not absorbed by the plants to be discarded through the drainage system, which has a little tube that goes straight to a tipping bucket. This tipping bucket helps us measure the volume of water coming out of the trays. These are my plant coverage methods. So I am using a 2 by 2 foot plastic quadrat that is used to visually sample 4 equally spaced out sections within each tray. Eventually, I would like to take full photos of these trays and process the images through an image processing program called ImageJ. This will help me figure out the leaf area index. In this case, it would be the ratio of the black pixels or the ecosystem, the plant canopy to the white pixels or the ecosystem. Here are some results for my plant coverage. I took monthly samples, which is mentioned on the x-axis. The green bars represent the buffalo grass structures, while the brown represent the white stone crop structures. You can see that in the beginning, buffalo grass is significantly lower in percent covered compared to white stone crop. However, over time, it nearly outcompetes the white stone crop by August. In general, the trend here is that plant coverage increases over time during summer. Some results for weed collection. This time I took bi-weekly samples of weeds starting in later August. The y-axis here is the average weed count. And you can see as well that the buffalo grass structures have a significantly higher weed count compared to white stone crop, except for September 13th, which is a very interesting result that was unexpected. In terms of white stone crop, generally there's just a trend of weed count increasing over time. So some conclusions and further implications. So today what we learned is that plant growth does influence the weed count throughout summer, as we saw with the relationship between the plant cover increasing as well as generally the weed count increase as well. And eventually I would like to identify most of the species that I can from just taking pictures of what I find within the trays and find like a difference between them, if there is any. And the main implication here is to provide plant data for the future clients that will be entering the Central City 2035 plan, which is a plan that where basically any new commercial infrastructure of a certain size will be required to implement an eco roof on top. Here are my references. And I would like to acknowledge Dr. Alyssa Starry, the Home Ecology Research Lab, Build Excedo, and National Science Foundation for the funding. And of course, my family and friends for the support. Thank you so much, and I can take any questions. Yep.
Sometimes feels like weeds come from nowhere.
Yeah.
Was your substrate weed seed free?
For eco roofs, it is required that it is weed free. However, there's a lot of animals like grasshoppers, for example, visit these eco roofs, bumblebees, wasps, just a lot of insects and also birds as well. I've seen a squirrel almost climb on top of these. So weeds can come from anywhere, but most of the time I'm pretty confident it's just coming from the animals that visit since it's an open system.
Where are the trays located and what is the amount of sunlight in that location?
It's the Walk of the Herons. It's right between the football field and SRTC. And the amount of sunlight that they receive, it's mainly just as much as possible.
Okay. Like emulating a roof.
Yeah, basically, that's the point, is to emulate the roof and have as much sunlight come down. However, there's some trees that do provide shade, but it's just like based on our like constraints.
Yeah,
we try our best. So.
I noticed on the slide where you have the comparison between the two species, that the last one, the stone crop went down. Was it like dying off from just the season or weed competition or what?
For plant coverage.
Yeah, because it was less in August than it was in July.
Yeah, so that could be because it is dying off. I've noticed that sometimes over watering the stone crop, because we typically water the plants like a few times a week, if not daily, during hot temperatures. And I noticed that the white stone crop doesn't really like a lot of water, so it started dying off just a little bit. So that could be an explanation for why there's less cover.
What exactly is the concern of weeds for rooftop gardens? Is it just like to limit the weight and like heat tolerance of the roof? Or what
Yeah,
is it?
so weeds can also displace the plants that were initially put there. A lot of people just like it if they have a monoculture of just like one plant. So if they see that weeds are displacing the plant that they intended to have there, it can be like visually unpleasant to see, or they just don't want the weed to mess with any like anything with the original plants. So just like nutritional benefits as well, sometimes they can take away from the system and then cause more plant die off.
They don't want to see something they don't like on their roof.
Yeah, it's very like, yeah, sometimes people just don't like seeing certain things, so it's like it just depends on like the goal of the consumer.
So it's not a concern about the health of the rooftop garden?
Sometimes it is, but a lot of people don't really look into like the health of the plants, which is also what this research is planning to provide, like more in-depth research on the plant species and whether they actually are bothered by certain weeds or whether it's okay to eventually leave them there and lower like maintenance costs, because that's like a common complaint among eco roof owners, is that they just don't want to pay extra for maintenance, as essential as it is.
Have you ever heard stories of people finding trees accidentally starting to grow on green roofs?
Yes, that
Can you
happened.
imagine with a tree root?
Yeah, I think that happened with me. I found a little like tree, but I couldn't identify what species it was, but I was pretty confident that it was a tree. But I know that there's plenty of roofs that have trees on top of them, except they have to be able to support the weight and also just be able to actually keep the tree alive over long periods of time.
That's pretty fast. Is there any other location you're thinking about setting up some of these trays on campus? Maybe like on top of a building or?
Unfortunately, this project is just meant for only the 7 trays that we have, but that would be very interesting to kind of like make these trays on a different side of campus and maybe compare like how the species grow on one side of campus compared to the other. Any other questions?
Am I allowed to ask one more even though we're over time? Great presentation. I have a bunch of questions, but I'll try and stick to one. For the weed data, were you also seeing those in your negative control, and did you subtract them from your data here?
Sorry, could you explain it?
So you had your negative control plot, so no other planting. Did you see weeds in there? And if you did you subtract them then from your experimental plots, as it were?
Oh, no, but I did not include, but for September 13th, I actually found 7 weeds in the control, which was really interesting because prior I couldn't find any weeds in them. I just didn't include them for the plot, not to confuse everyone.
Interesting.
One more, because you didn't get to this. So what's happening with the stormwater data, with the runoff data?
Yeah, so the stormwater data is going to be used to compare to the plant cover data, and we're eventually trying to compare the species and how much runoff is coming from these trays as the plants grow to their full coverage.
And who's collecting that data?
That's from the environmental engineering people. Led by Dr. Samantha Hartzell. Right here.
So how long does it take for these species to have like full coverage? Will they eventually cover the whole?
Yes, that's the plan. Typically, it depends on the size of the roof, but usually it takes about a year or 2 to fully establish. We're hoping it will be full within the year because that's when my thesis is supposed to be complete. So hopefully they grow fast enough because they've been a little slow. We've had to add like more plant cuttings and seeds just to populate the roof more because we started a little bit later than expected and they just were not growing as fast as we wanted to. So to promote them, we just added more.
Just a real quick follow-up though, how do you start seeding? You started at like a 10% coverage level, or what's the general start position?
I did not, I should have, but I did not like calculate the percent cover for the start, but I believe it was about maybe like 30% for the white stone crop and then about maybe 20% for the grass. It started really tiny and now it's just spreading all over.
Like 3 inches on
So,
center or something.
Now we will have to, everybody, I'm pleased to announce our next presentation, and I just want to make note that this presenter is in the URISE program, our newest cohort, and just was placed in the research lab this summer, and I just heard this reflects about 2 weeks of time in the project. So I'm pleased to introduce Sofia Villarreal, who's been working with Dr. Anna, and I'll get to turn to macaques. So thank you.
Thank you, Dr. Keller. So good morning, everyone. So this summer, I work with the Oregon National Primary Research Center, as well as ARCOR, to assess rhesus macaque sperm before and after freeze drying. So non-human primates are an important asset in research projects in which investigators require specific genome customizations to be done with their animal models. And to address this ongoing demand of NHPs, cryopreservation methods like liquid nitrogen are needed in order to store the sperm's DNA. And while liquid nitrogen is efficient in doing so, it does come with a lot of drawbacks in regards to its handling, its storage, and its cost. So alternative preservation methods are needed in order to address this problem. So freeze drying or lyophilization is a type of method which basically removes the water found in biological components through a three-step process. First being freezing, primary freezing, and secondary freezing, so that it can get into this solid-like state that's easily able to be transported and refrigerated. And while the objective of the study was to collect fresh and freeze-dried sperm and evaluate them using 5 different assays in order to look into their DNA, acrosomal, plasma membrane, and mitochondrial integrity. Study design methodology consisted of 3 different phases. The first being preparing and collecting the fresh and freeze-dried sperm samples so that the 5 assays, tunnel, FITC PSA, simple acrosome, live dead, and mitotracker orange could be done in order for the damage assessments that look into different structures of the sperm. So DNA fragmentation, the acrosome, the plasma membrane, and mitochondria can be looked into how much they were damaged before or after freeze drying. And so for the first tunnel, this looks into the DNA fragmentation, and we can determine how much DNA fragmentation or DNA damage done looking at the head of the sperm where the DNA is found. So depending on the brightness, that determines how much damage was done. The next 2, FITC PSA and simple acrosome, look into the acrosome layer that is basically on the sperm. Sperm's head is like a layer filled with enzymes that help during fertilizations. And for example, with FITC PSA, we can see if it was damaged or not, depending on the brightness of the grain. So the bright green means that the plasma or the acrosome was intact. With the simple acrosome, it plays similar, but with different colors. So with having a lavender finish at the end, that determines that it was intact. Live dead looks into 2 different stainings. So live meaning that the plasma membrane was intact, and it uses a green finish shown as down there. And with the dead, it looks if the plasma membrane was damaged. So it will make it into an orange or red color. Mitotracker, it doesn't show, but mitotracker orange, like it says in the name, looks into the mitochondrial activity found within the mid-piece of the sperm that helps with motility. And if it's orange, it shows that it is active, and it's not, it won't show any orange in the mid-piece. So results and conclusion. So our results shown in the bar graph where we collected the positive percentage of sperm. So for tunnel assays, we found that there was more DNA damage found within the freeze-dried sperm in contrast to the fresh samples. And with the other 4 assays that look into the acrosomal, plasma membrane, and mitochondrial damage assays, we found with all of the freeze-dried sperm, we found there was non-intact acrosome, non-intact plasma membrane, and non-active mitochondria, in contrast to fresh sperm, where there was some that we found that were negative, but not as much as the freeze-dried sperm. So overall, these findings are really important towards moving on with determining whether freeze-drying can be an alternative for preservation of NHP sperm. And these findings also help validate the assays do in fact work and can determine whether the part of the sperm structure was damaged or not. And we can start enhancing these freeze-drying techniques, as well as starting to attempt to do fertilization methods like ICSI. I want to thank a good shout out to the ARCOR team, who without them this project would not have been able to do. And I want to thank you all for listening and wanting to learn more about NHPs and their preservation methods.
Wanted to know, how do you resuspend your lyophilized pellets, and what process that is?
So you're asking about the process where we the sperm process and very beginning, how we rehydrate it, or are you talking about how?
Opposite. So like after they've been freeze dried, like how do you resuspend them so you can analyze them?
Yeah, so once they've been freeze dried, this is really simple actually. Since inside the sperm, in that little solid state, there's media that we just rehydrate with just molecular water or just distilled water, and then we make it so that it's completely dissolved. And then we just start doing the assessments after. It's really easy. So
cool.
Yeah.
I might have missed this. I don't think I did. Does time play a major factor in the damage? Like what's the length of time you guys are allowing these to be freeze dried and then settled, or seeing how much damage things have? So
I think time should, that's a good question, because with the freeze-dried samples I worked at, I think they were several months old. I think it was around 3 months. Since we just started doing these experiments with these freeze-dried germ, we don't really know how time would play, but that would be a good question, I think, to later on evaluate, I think. Yeah.
Yes, curious, from what I've heard, lyophilization is as much an art form as it is a science in terms of what you add, how rapidly you freeze, et cetera. So are you thinking about, this is kind of what Marcel was talking about, when you're resuspending, say it's in a media, have people thought about modifying that media somehow so it might work better? The things you have in the media before you do the lyophilization, not the sperm, but everything else which is in there.
I think we probably could, because like I said, we just did this, so we're trying to see how we can enhance this, because for example, we weren't expecting certain outcomes, for the acrosome, we weren't expecting it to be not intact for all freeze dried. Right. So that's something we need to enhance and look into in order to make it much more reliable for preservation. So we could adjust the media or do some sort of alternative technique while after freeze drying.
Yeah, all these different tests that you do describing whether or not it works, are some better than others? Do you think you need moving forward to do all of these tests, or do you think one is better for specific purposes than the other?
I mean, yeah, we did, for example, the acrosome, we used 2 different assays, and we saw that we preferred looking at the simple acrosome because it was way easier to identify whether the acrosome was intact or not. And for example, tunnel, when I was doing the essays and looking at calculating all the positive sperm found, it was a lot of work to do counting, but it is the most effective in looking into the DNA fragmentation done. So I think there's probably preferences, but this was mostly just to see if they looked good or not and how we can determine their damages, but yeah.
Interesting trade-offs.
How do those defects affect the viability of the sperm? Is it still like moving normally?
Yeah, so that's interesting, because I asked the same thing to my PI, because we saw like all the freeze-dried sperm, in contrast to the sperm, you would think these are not usable. But what matters is the DNA found within, because at the end of the day, ICSI, I didn't really talk about it, but ICSI is basically a process where you like move with the egg, you move the sperm inside the egg. It doesn't have to have any motility whatsoever. So what matters is having that DNA good, like it's able to fertilize at the end. So it did show like the DNA was soil damage, but it was not as good as like a fresh, but it should work. But what matters is the DNA found in the sperm.
So they still move?
No, it doesn't move, because the motility, the mitochondria or the mid-piece, that helps with the.
Yeah, like you don't need it to move.
Yeah, so if we're doing it, we don't need it to move. In contrast to in vitro fertilization, where it does require motility of the sperm. So. Thank you.
Another one. So actually, I was also curious a little bit about your tunnel stain. You had, your positive, your negative in your test. I wasn't entirely sure which was which there. So, either here or in your
Yeah, so for positive,
yeah.
it's that one over there, and basically all the positives, those are the ones where we identify how much damage was done, like, because the tunnel uses an enzyme that breaks into the DNA, and depending on how much damage, it causes it to be brighter.
Right.
So we saw that positive, we found that there was more DNA damage done in contrast to negative, where we're expecting no DNA damage done, and tests we did find some. So.
And so for the DNA damage, did you actually damage the DNA there? So how are you getting that?
Yeah,
So,
we're damaging it using, for positive, we would use liquid nitrogen because for positive and test use because tunnel like I said, uses an enzyme and a label.
Right.
So positive and test contain that enzyme and label, while negative, but the thing is positive uses that liquid nitrogen, which is at sub-zero temperatures, which damages that DNA. So
it's damaging the DNA. Isn't that what people were using now? Those are the idea that.
So the thing is with like liquid that uses a cryoprotectant
Okay.
beforehand. So there's a whole process to because obviously if we just submerge like with media and with sperm, we just submerge it with liquid nitrogen, it's gonna damage it. But it uses a cryoprotectant called glycerol.
Yeah.
We use it. So
And so, okay, that's the difference. Okay,
Yeah,
sorry,
that's the
I
difference.
missed that.
yeah,
that
no.
difference. Okay.
Yeah.
And then the negative is just again without the enzyme or.
Yeah, without the enzyme and without the liquid nitrogen.
Without doing the liquid nitrogen, yeah, so you're still seeing some fluorescence
There's
there.
still some, which it was like,
Yeah,
you
so
know.
you're looking at the difference between the two. Cool.
Yeah.
I think we'll just get started then on our last presentation. And I'm really delighted to introduce Luann O'Connor, who is a scholar who started Exedo at American Samoa Community College and is now working in Dr. Stedman's lab. And we'll let her begin the presentation. Thank you.
Good morning, everyone. My name is Luann O'Connor, and I'm a member of the Stedman's lab, also known as the Extreme Viruses lab here at PSU. Today we'll be looking at the construction and analysis of VP1 mutants in our extreme viruses. So one of the viruses that we study in our lab is the Cephalopus spindle-shaped virus 1, which is an archaeal virus that is part of the Fusilobacteridae family. This virus consists of a circular double-stranded DNA genome, and it infects Saccharomyces solifitericus, which is its natural host, because every virus needs a host to survive. And this virus and its host both survive in extreme environments such as geothermal hot springs, with a high temperature generally above 70 degrees Celsius, with a pH level below 4, close to 0.9, which is very acidic. And there are four proteins that make up our SSV1 virion that you see here. And one of them we'll be focusing more throughout our project is VP1, which is our major capsid protein. This VP1 major capsid protein works together with our VP3 minor capsid protein to form the outer structure of our SSV1 virion that you see here. Our major capsid protein VP1 consists of a cleavage site, glutamate E at position 66 of VP1. This glutamate is an amino acid that's highly conserved throughout all spindle-shaped viruses. And the mutation was made at this cleavage site where we substituted the wild-type glutamate, amino acid glutamate, with other amino acids. And as you see here, it resulted in 2 mutants that had significant differences in capsid size by length and width. As you see here, our glutamate mutants, these are its virions, and it had a significant difference in capsid size by length and width compared to our wild-type glutamate virions that you see there. However, our isoleucine mutant only had a significant difference in its capsid size by length of its virions compared to our wild-type that you see there. So what we did, we made constructs within these additional SSV1-VP1 mutants, introducing 6X his tag, which is a purification tag. This was done in order to observe the levels of VP1 expression and for it to identify VP1 and SSV1 virion and allow purifications to take place. So how we did this whole process is that we have our EAA2D3 plasmid, which is our wild type. We introduced the 6X his tag, exponentially amplifying it. And then we have our 6X his tag attached to the ends of our fragments there. And then we utilized the KLD treatment where we got our newly synthesized plasmids. We transformed it into E. coli, and then we played it on LB and kanamycin to get our colonies, took it out for our, and then we extracted the DNA. We then sent it out for sequencing using one of the VP1 primers, and then we electroporated it into our host S441 Saccharomyces ophiotragus. As you see here, we did the supernatant PCR followed by halo assay, and then we sent it out for sequencing again. And then we took it to the TM microscope at RLSP to scan or for mutant viruses. As you see here, this is a picture of our sequence that we went for the 6X his tag. Highlighted is our 6X his tag sequences, and our histidine mutant on top in our wild type had the 6X his tag purification tag. However, our tyrosine and aspirin mutant did not have it. And before all of this was done, we did a mutagenic PCR where we introduced the 6X his tag. And as you see here, these bands were shown, showing that it indicates that the 6X his tag should be in all of these mutants. However, from our sequences here, only 2 had the 6X his tag. So we continued on with our 2 mutants, and then we electroporate them into Saccharomyces ophiotragus, which is the host. And then that was to grow viruses. And then we did the supernatant PCR. This was to test for viral DNA in our glutamate wild type that had the 6X his tag with the histidine mutant. You see there's a faded band. So, I mean, there's a band, so it should indicate that there are viruses, but our histidine mutant, however, did not have a band, so there was no viruses. Here's a picture of our VP1 alpha predicted structure wild type compared to our VP1 with the 6X his tag. The 6X his tag should be right around here as you see compared to our VP1 wild type. The difference is that this one has more, that's the name of it, compared to the wild type, which is these squiggly things. And then highlighted in red is our glutamate position 66 cleavage site. For our future directions, we are to further mutate our histidine and tyrosine mutants you see there. That's the picture of the virions taken at the TM microscope. That was before, these are mutants without the 6X his tag. So further mutations to be done, introducing the 6X his tag. We made a mutagenic PCR introducing 6X his tags to our mutants that were significantly different in capsid size by length and width. As you see here, we have our glutamine and isoleucine mutant. So we are to perform the KLD protocol where it will phosphorylate the ends that has the 6X his tag, and then for it to get our newly synthesized plasmids. So that's to be done. We will work on mutants that did not have the 6X his tag with using different primers. And we will repeat the electroporation with the mutants that have the 6X Hesek and scan for viruses, because we believe that's the process where we kind of wasn't producing viruses. So we will also follow by Halo assay protocol, that is to test for the infectivity of our mutants. And we'll continue the process by making protein gels. This is to observe the levels of VP1 expression. These are my references. And I'd like to take the time to thank my PI, Dr. Stedman, and my mentor, Jono, and the whole Extreme Virus Lab for the support and opportunity to work on this project. This was a picture that was taken last month at Last in California. We were on our sampling trip. And also, all these organizations for funding this project and the support, and for my sisters and family back there, and for being here. And if you have any questions, please let me know, or you can scan the QR code that will take you to the Extreme Virus website, and you can learn more information about our viruses. Yeah.
Questions? Speechless.
What's the host called again? The host is Saccharomyces sophiatricus.
Is that like a bacteria or like an actual like?
It's an archaea.
Oh, archaea, okay.
Yeah, and as you see here, I believe these are the host cells, and this is the picture of our SSV1 virions that are infecting it.
Okay, thank you. What has been your favorite part of the protocol, since it's so lengthy?
The protocol, I believe it's wait, we'll go here. Well, to me, it was the colony part. Fun picking the colonies when you have colonies. Yeah. And also getting the sequences where the 6X Hesek was successfully attached to some of our mutants. Yeah.
What part of the experiment did you have to repeat the most? 'Cause I've always had trouble with transformation.
I had trouble with transformation as well, like doing the colonies, having colonies. But I got lucky one time. I had to repeat it 3 times. Yeah, and then another thing was that I'm thinking about is electroporation. I repeat that again too because I wasn't producing any viruses in my mutants. Yeah.
Right, well, thank you again for a wonderful presentation. So, again, really impressive work from all of our presenters. I wish I could say I learned a lot, but I don't know if it's all gonna stick. But just really great work over the summer and longer in many cases. A great combination, and I hope that a lot of these projects turn into conference presentations and publications. Now you've had some practice presenting, and again, I think from my perspective, those were all conference worthy presentations, so congratulations.